Welcome to RGR Academy. This is biology session. We move to the next chapter, that is cell cycle and cell division. We'll discuss the questions and answer, and we move to the justification part of this particular chapter. Alright, the first question 91. The checkpoint in cell cycle plays an important role. Out of the four options given, out of the cell cycle we are talking about, the assessing the DNA damage, because this is the particular time where the preparation phase we call it as an interphase. In this particular phase where all the preparations for the cell divisions usually takes place, not only the division of the DNA, that is the copies of DNA is about to get increased in case of like synthesis phase, but also you could see the preparation for like the damage. If there any particular damage which seems to be occur in the chromosome or in the gene or in DNA, like that has to be rectified in this particular phase. So the question says the checkpoint, there is a checkpoint that we could see, right, because we have mitosin, the checkpoint 1, checkpoint 2, checkpoint, there are 3 checkpoints that you would have studied, right. So out of this, where you could see the checkpoint in the cell cycle, where this checkpoint is for the assessing the cell damage. If there is any damage that has to be rectified in this particular phase itself, if this damage, the proofreading mechanisms, like if it's not happening in this particular phase, eventually this cannot be rectified, then the damage would have been continued to the next phase, I mean to the mitosis, the cell division, where again it leads to the production of like it's an incompetent cell. So for this particular question, option 3 is the correct answer, assessing the DNA damage. Question number 92. The checkpoint in cell cycle plays an important role. Again the same sort of questions. Whenever any question comes like checkpoint, so what for the checkpoint? The checkpoint is to check whether is there any damage which present in the cell. So for that only we have the checkpoints, the 3 checkpoints that we have, like one we have the metaphase stage also. So this checkpoints will be very keen in checking is there any damage in the cell. Next question 93. Some cells in adult animals do not divide, they exit G1 phase and enter into an active stage. So what this particular phase where it will not divide, it will go to the something a resting phase where it doesn't mean like it's metabolically inactive, it is metabolically active. But what is that phase that we call it as? We call that particular phase as a quiescent phase. For question number 93, when we draw the cell cycle, we are talking about, if you done with this part already, we talk about this part. So this we call it as an M phase, this we call it as an M phase, and this part is called as interphase. This part is an interphase, we call it preparatory phase, we call it as an interphase, in which you could see here we have like the G1 phase we have, and we have this G1 phase we take, and this is the synthesis phase, and this is your G2 phase, and in between you have something called as the GO phase, which occurs, a very small phase which occurs here, even I can make it very smaller, like this phase, the smaller phase, this we call it as an GO phase. This GO phase is also called as quiescent stage or the quiescent stage, where what happens like in the prophase you would have studied like the M phase, prophase, metaphase, anaphase, telophase, cytokinesis, prophase, metaphase, anaphase, telophase, cytokinesis, all these things takes place in the M phase. But one point of time what happens like if there is any damage which occurs in the cell, for example the cell is not capable of going for a cell division for the next cycle, then definitely the cell will go to the quiescent stage, that is a gap that you could see called GO, this called as a quiescent stage. In this particular stage where what happens like there is non-availability of the mitogens. So the cells which is going to this part right, these cells we call it as an reserve cells, these cells we call it as reserve cells, and these reserve cells where then there is no availability of the mitogens are not present in this particular phase, so no mitogens are present. So it doesn't mean the cells are metabolically inactive, the cells are metabolically active, but any point of time this cell they can resume back to its own cell cycle that can occur. This you would have studied in case of like differentiation, redifferentiation and dedifferentiation, that mechanism also. So for this particular question, option 2 is the correct answer. Question number 94. S phase of the cell cycle. So I drew already like what's G1 phase, what's the S phase, and this is a G2 phase. G is a gap 1 phase, this is a synthesis phase, and this one is a G2 gap 2 phase. They are talking about the S phase. The question they ask is what exactly happens in the S2, that is a S phase? The amount of DNA doubles in the each cell. So here what happens? The amount of DNA. So the amount of DNA which gets doubled, the amount of DNA gets increased, it be doubled. So this is the place where you could see synthesis. But this time, this phase in G1 phase that you could see, there's no increase in amount of DNA. There will be only increase in the size of the cell. Size of the cell is getting increased, but not the DNA. The DNA content remains same in the G1 phase. But in this phase, the S phase, the synthesis phase, where the amount of DNA is getting double. Where you could see now, the 1C will be converted to 2C, right? Whereas in this case, like 2C will be converted to 4C. This in case of haploid cells where the 1 chromosomes won't be converted to 2 and 2 will be converted to 4. This is in case of diploid cell. This is in case of diploid cells where 2 cells can be converted to 4 cells. Fine. So for this particular question, option 1 is the correct answer. Question number 95. The centrosome duplicates during. So when the centrosome duplicates, it duplicates in the, because the DNA is getting double. So DNA is getting double, of course, like the definitions like it says, like where you could see the S phase of the cell cycle, where in this part the interphase of the cell cycle, where the centrosome are divided into 2 centrosomes. So these 2 centrosomes are getting formed where in this case of the S phase. So for this question 95, option 2 is the correct answer. 96, DNA content is doubled. They didn't ask about any particular phase within the interphase. In interphase only we call it as the together. G1 phase, S phase, G2 phase together we call it as an interphase. In specific they ask is whether it is an M phase or in an interphase where the division of the DNA is getting doubled. Where DNA is getting doubled? The DNA is getting doubled only in the interphase. So for this question you take 96 question. Interphase is the correct answer, where the interphase is the correct answer, where the DNA is getting doubled. Especially where in the interphase, when more specifically if they ask in interphase is a synthesis phase. Or you go with the M phase or the interphase, then the division of DNA which is occurring only in the interphase, that in synthesis phase more specifically. Okay. So next question 98. Amitosis, the word which comes definitely you go with the bacteria. The bacteria only you could see this type of process where amitosis occurs. Amitosis where there you could see no differentiation of the chromosome and spindle. So in this case where you see 98 question, amitosis where there is no differentiation. Usually you could see in this case of bacterial cell, no differentiation usually, and spindle formation. I'll write again here, I'm sorry. So spindle formation occurs, no spindle formation. Where this usually you could see often in the bacterial cell. You could see in a bacterial cell. So this is not a regular method of division because the cells do not go for like dividing the nuclear matter equably, because some authors they represented like they include the division of Monera. The kingdom Monera, right? Our 5 kingdom concept you have studied. The kingdom Monera, which usually goes with the process called as amitosis. So for this question, the correct option is 1, bacteria. Then question number 99. Which of the following is not a characteristic feature during mitosis in somatic cells? In somatic cells, which is not the characteristic feature? You could see the chromosome movement, you could see the spindle fiber, you could see the disappearance of nucleolus. But one thing that you could not see in the mitosis in the somatic cells is a synapses, because synapses is a feature that you could see right before the crossing over, where the synapse, the dead form of the chromosomes, homologous chromosomes get arranged only in a meiosis, not in a mitosis. So for this the correct option is 2. 99 question, option 2 is the correct answer, because in meiosis only you study like diplotin, pachytin, zygotin, all the stages, substages that you would study in the prophase 1, but this you could not see in a mitosis. Question 100. During the metaphase stage of mitosis, spindle fibers attach to the chromosome at where this exactly attached during the metaphase. When you talk about the metaphase stage, example, you take, if this is the cell we are talking about, here you could see the centriole and this is the ester formation. Centriole, you could see the ester formation, and here you have the spindle fibers. Here are the spindle fibers that you could see. The spindle fibers which start arising over here, and here you could see the chromosomes that you could be lined up that you could see in the equatorial region. This we call it as a metaphase plate or equatorial plate. So where exactly the spindle fibers which comes in bind onto the disc-like structure, you could see the disc-like structure which had adhered onto the surface of what? Surface of the centrosome. This structure we call it as the disc-like structure which present onto the centromere. This we call it as kinetochore. Onto this only this spindle, the spindle fibers which comes in bind. So what happens, this will be pulling the spindle fibers which pull the chromatids into two equal halves on both the sides, so that what will happen, one set of chromatids will move towards this pole, another set of the chromatids which will be moving to the opposite. So what happens, the divisions, the one half will be going this side, the other half will be going on the other side. How this has been pulled? This has been pulled by means of the spindle fibers get attached with the disc-like structure onto the centrosome, that we call it as a kinetochore. So the correct option is kinetochore. Question number 101. 101, a stage in mitosis that starts towards the middle of the anaphase and it's completed with the telophase. They've got the stage that we call it as when we now talk about the M phase, prophase, metaphase, anaphase, telophase, and the stage that we are talking about here is cytokinesis. So for this particular question, the correct option is cytokinesis. This is for question number 101. Option 1 is the correct answer. Then question number 102. At metaphase, chromosomes are attached to the spindle fibers by this again the same question. During the metaphase stage, this we are talking about the metaphase stage. During metaphase stage, we could see the equatorial plane or the metaphase plate has been formed. This spindle fibers is getting attached with the structure that was called as a kinetochore. So this we have done already, this particular question. So question number 102 is also the same answer, kinetochore. Option 3 is the correct answer. 103, spindles are arrested by colchicine. Colchicine is actually the alkaloid derivative that you use actually. This particular substance which stops the cell cycle, and especially at the metaphase stage, because metaphase is an ideal stage where you could see the structure of the chromosome very clearly. You would have studied about the karyotyping, the ideogram techniques by which we can easily identify the structure of the chromosomes or chromosomal abnormalities, chromosomal aberration, or in case of gene therapy, any genetic disorders where you could see where you want to look at the microscope structure very clearly. For that, it requires, you need to freeze it actually. You need to freeze that cell cycle. Which phase especially? You need to freeze it at the metaphase stage. What is the substance, the derivative that you add here is colchicine. Colchicine is actually the derivative that you are supposed to add. So that what happens, the spindle fibers are arrested. If the spindle fibers are arrested, how the spindle fibers are arrested? By adding a alkaloid derivative. This we call it the colchicine, so that you can stop it, so that it cannot be pulled over onto the opposite poles, so that it will not go to the next stage, that is prophase, metaphase. It stops with the metaphase itself, it will not go to the next stage, that is anaphase will not occur, so that you can look at the structure of the chromosomes very clearly. So for that you use colchicine. So for this particular question, option 2 is the correct answer for 103. Question number 104. Spindle fiber is made up of, so what is a spindle fiber? The spindle fibers, it's made up of actually tubulins which are microtubules, through which only the microtubules are formed. So question number 104, tubulin is the correct answer. Tubulin is the correct answer for this one. Question number 105, best material for the study of mitosis in the laboratory. Laboratory, the best material is, instead of anther, leaf tip, or you go with the ovary, the best thing that you can study is the root tip. Root tip, where you go for the meristematic tissues, it's the best thing that is a choice. If you want to select, then you go with the root tip when you go for in vitro or lab technique studies. So we're done with 105. 106, chromosomes are visible with the chromatids at this phase of, in which phase the chromosomes are clearly visible. So this we already discussed. This is also for question number 106, metaphase. Again the same sort of question, only in the metaphase you can see the chromosome structure very clearly. In meiosis, crossing over is initiated. So when we talk about the meiosis, as you know, mitosis is an equational division and meiosis is a reductional division. Where meiosis you could see N, haploid, you could see in the germ cells. right? So for question number 10, yeah, okay. So question number 107. As I told you, like this is a basic understanding where you could see mitosis which gives to equational divisions, where you could see like 2n, where in meiosis is a reductional division, you could see only n. This occurs in a somatic cells. This occurs in a somatic cell, where this occurs in a germ cells by means of gametogenesis, like spermatogenesis, oogenesis, where this process which occurs. So this meiosis is a reductional division. This one is a reductional division, and mitosis is an equational division. This is an equational division. So when we talk about the meiosis, what the question they ask is like the crossing over. So when this crossing over initiated, in which stage? Prophase 1. In the prophase itself, it's a very lengthy phase in the meiosis, that one that you could see, leptotin, diplotin, zygotin, pakitin, that you keep on discussing. The lengthy phase that you could see in meiosis, one that is prophase 1. Under prophase 1, there is a stage called as Pakitin stage. In this Pakitin stage, only what happens is homologous chromosomes which form the structure. This is homologous chromosomes, and these homologous chromosomes which form the structure. This is another homologous chromosomes, for example, you take. Okay, this is another homologous chromosomes. This structure which is getting formed during the Pakitin stage, and this we call it as a synapse formation. This formation, the dyad, we call it as this dyad-like structure, the dyad structure which forms, this during the Pakitin stage, the synapse formation. So correct option is 107. That is the third one is the correct option. 108. The stage 2 meiotic divisions is called, the stage between the two meiotic divisions, like a small gap which takes place, no division, nothing will take place, there's a small gap, the breathing time which takes place, right? That we call it as interkinesis. Interkinesis, that is a stage where two meiotic divisions in gap, that is a gap that you could see. So option 2 is the correct answer. Crossing over occurs in which stage? Again in the Pakitin stage. Crossing over which occurs in the Pakitin stage. So question number 109. You take the question 109, crossing over. If this is one homologous chromosome you're talking about, right? This one homologous chromosome, and this is like, the homologous, the other chromosomes, where you could see, this is get, entangled at the surface of the non-sister chromatids. This is one chromosome and this is the other chromosome. So here you could see the crossing over takes place between the non-sister chromatids of the homologous chromosomes. This also which takes place in what? Takes place in the Pakitin stage, and this we call it as a crossing over. This leads to a recombination by means of an enzyme called as recombinase enzyme. This is a characteristic feature that you could see in a meiosis in prophase 1, that is in Pakitin stage. Question 110. The enzyme recombinase is required. I told you again, like question 1110 also. Question 1110, for this process to occur, because this has been attached together. These two chromatids been attached by means of synaptonemal complex. This been attached by means of synaptonemal complex. So then what happens due to the recombinase enzyme? The recombinase enzyme which leads to the formation. Now this requires an enzyme called as recombinase enzyme. This recombinase enzyme which mediates this process called as crossing over. So question 1110, this also occurs in the Pakitin. So Pakitin which plays a very significant role as far as like crossing over, the synapse formation, all those things too takes place, right? So Pakitin. Question 111. Statement A, for the particular character in an individual, each gametes get only one allele. Statement B, chromatids of a chromosome split or separate and move towards opposite poles during anaphase of mitosis. Here the option is both the statement are correct, but B is not the reason. B is not the exact reason, that is for A. The reason is because the statement A is a Mendel's law of segregation. The statement shows like it's a Mendel law of segregation, but which is a universal without an exception. During anaphase, what happened? Chromatid separates and move towards the opposite pole. So both are correct, but B is not matching with the A. So for that we go with option 4. Next 112, a bivalent of meiosis, it occurs in a bivalent of meiosis, one which occurs in 4 chromatids and 2 centromeres. Bivalent is what? Here you could see 2 centromeres and 4 chromatids. 2 centromeres equal 4, 1, 2, 3, 4. This 4 chromatids that you could see, this is a structure of the bivalent. Okay, so 4 chromatids and 2 centromeres that you can see. Meiosis 1. Question 113. During gamete formation, the enzyme recombinase participates during, I told you like the packitine, all the stages which comes under what? Comes under the prophase 1. This is a part of what? This is a part of prophase 1 of a meiosis. Under prophase 1 only this packitine stages come. So, prophase 1 is the correct answer for this particular question. 113. 3 is the correct answer. 114. During cell growth, DNA synthesis takes place. I told you already where the DNA synthesis, that's the reason we call it the synthesis phase. This S phase is called as synthesis phase, where the DNA content is getting doubled here. This we already discussed. This is for the question number 114. If you are very clear with the cycle, the interphase and mitosis, you can answer any type of questions which comes from the cell cycle. For that, you should understand what is interphase, what are the substages of interphase, what are the stages, substages in the M phase, that is in mitosis, and what is the GO phase that you should remember. So, for this question, 1 is the correct answer. Next question 115. Which one of the following does not occur in the interphase of the eukaryotic cell division? Interphase where the reduction in cell size, but in all the cases where you could see there will be increase in the cell size, whether the DNA content is increasing or not, but the cell size which keeps on increasing, there is no doubt in that. So, option 4 is the correct answer for this one. Next question number 116. In a diploid cell, at which stage of cell cycle amount of DNA is double? Again the same question, S phase. In the S phase, this is also for the question number 116. In the S phase where the DNA content is getting doubled. We already discussed. Option 4 is the correct answer. Doubling of the chromosomes is termed as. So, this doubling of the chromosomes we call it as a duplication. We call it as duplication. If the what is it called, replication, the term comes, you go with the replication, or else the duplication comes means you go with the duplication, because it's same like a replication. So, 117 option 1 is the correct answer. 118, DNA replication occurs during which part of cell cycle? Again the same question. 118, see how many times they ask the same questions, are only in the synthesis phase where the DNA content gets increased. So, option 3 is the correct answer for 118. During which phase of the cell cycle DNA duplication occurs? Say they twist and ask the same questions a different way. For this also the correct option is 119, also the synthesis phase. So, repeatedly they are asking about this particular phase where the DNA content is getting doubled only in the synthesis phase. Question 120. Select the correct statement related to the mitosis. If you read all the statements where 1, 3 and 4, all the statements seems to be correct, but the incorrect, where one statement that you could see, the amount of DNA in the parental cell is first doubled and then distributed into the 2 daughter cells. This is a very simple concept where first the karyokinesis takes place and then only the cytokinesis should take place. So, first the division of the nucleus, that we call it as karyokinesis. So, question number 120. 120, if you could see the cell, if there is a division of the, first you could see the division of the nucleus, that is division of nucleus we call it as karyokinesis. This should occur first, and then followed by the groove like structure should be formed. Then it comes to the next stage, that is called as cytokinesis, where there will be division of 2 cells. The division of 2 daughter cells will be formed. So, 2 daughter cells getting formed by this process. This is called as cytokinesis, where there are certain exceptions that you could see in the case of endosperms, where it's not a regular flow, where the karyokinesis is not always mediated by the cytokinesis, where the first cytokinesis takes place, then the karyokinesis. Then what happens in that case? More nuclei will start accumulating inside. If more nuclei start accumulating, this leads to formation of syncytium or multinuclear stage. In live examples where you could see in case of coconut or tender coconut, the endosperm formation, this is a classical example where the karyokinesis is not first taking place. First the cytokinesis is taking place, but in general situation mitosis, what happens? The first karyokinesis, division of nucleus, followed by the division of the cytoplasm, that is cytokinesis takes place. So, we are done with this particular question 120. Next 121. A stage of mitosis shown in the diagram, which stage is and where is the characteristics? I already told you this particular diagram. This is what they given in this particular question. Metaphase stage. The metaphase stage where you could see the structure of chromosomes very clearly. This we call it as a metaphase plate or the equatorial plate where all these chromosomes are assembled in the correct fashion. So we can see the structure of the chromosome in very clear manner. So where the colchicine and the alkaloid derivative that used so that it can freeze the cell cycle. So for question 121, option 2 is the correct answer. 122, select the correct options with respect to the mitosis. Chromosomes moves to the spindle equator and get aligned along the equatorial plate in the metaphase. This is the correct answer. So mostly you could see in case of like animal cells where we could see the smaller chromosomes which will be assembling the center, whereas the larger chromosomes which will be on the edges. But this is not quite slightly different in case of plant cells, because the plant cells you could not see the centrioles. Centrioles are absent in case of plant cells, and in animals you could see the presence of centriole. This makes a slight difference between the plant cells and the animal cells. For this question, option 4 is the correct answer. 123, arrange the following events of meiosis in the correct sequence. If you look at the sequence where the crossing over synapse and terminalization disappearance, where the fourth one is the correct answer, where synapses, then the crossing over, then you could say terminalization, then the disappearance of the nucleolus. Because first what should happen? The synapse should occur, then the crossing over, then terminalization should occur, then at last only you could see the disappearance of nucleolus. Question 124, match the items in column 1 with those of column 2 of the correct answer. With this, P is actually related with 3 and Q is related with the 4. So for this, option 3 is the correct answer, 124. Next we move to 125. The complex forms by a pair of synapse homologous chromosomes that I already told you. This we'll call it as the bivalent, where you could see the two centromeres, whereas you could see the 4 chromatids we could see. So for that we call this as a bivalent. This we call it as a bivalent. For the question 125, we call this as a bivalent. 126, the term synaptonemal complex refers to. So what do you mean by synaptonemal? I told you already about this particular part, synaptonemal complex, which forms between 2 chromatids of the homologous chromosomes, where it is responsible for the chromosome alignment. The chromosome should be properly fixed. It should form this type of dyad actually. It should form the dyad form. The dyad should be formed, and this must be responsible for recombination. If the synaptonemal complex is not formed, then the recombination cannot take place, the crossing over also cannot be influenced. So option 4 is the correct answer, chromosome alignment and recombination. Question number 127, visible expression of the genetic phenomenon of a crossing over. Visible expression, what is the visible expression that we call it as? X-like structure, we call it as X structure, where what happens, like one chromatids and the other one, like it will getting exchange over here, right? So this leads to formation of this other one. You could see this other one. So here you could see exchange of the genetic materials which takes place between these 2 homologous chromosomes. This we call it as a chiasmata. This we call it as chiasmata formation. This prominently which gives like X-like structure. So this X-like structure we call it as chiasmata. This visibly can be expressed out. So option 3 is the correct answer. Question 128. Which is the select the correct match? S phase. I told you already S phase is for the DNA replication. So 128 also the S phase is the correct answer. We discussed already. 129. Synaptonemal complex is formed during the which phase? When you talk about this phase, the synaptonemal complex, because it occurs before the Pachytene stage. But when you talk about the synaptonemal complex, when the synaptonemal complex which occurred before the Pachytene stage, that is zygotine stage. So after the zygotine only the Pachytene comes. So synaptonemal complex would have been formed before the Pachytene stage. So first is the synaptonemal complex, then only you would have seen the formation of the synapse. The synapse which formation occurs in the Pachytene stage. For the synapse formation, the synaptonemal complex would have been formed before the Pachytene stage itself. This we call it as zygotine stage. So first is zygotine, then comes the Pachytene. So for this correct option is 4.